Hello, my name is Susie Hoops. I'm a PhD candidate in Knight's Lab at the University of Minnesota. Welcome to my talk, Identifying Patterns and Addressing Pitfalls in Beta Diversity Analyses. I have nothing to disclose today. So to begin, I'll give some context for beta diversity analyses in microbiome studies, then present some current challenges in beta diversity analyses. Along with these challenges, we'll discuss beta diversity measures and identify patterns of similarity between different measures, as well as discuss the impact of compositionality. Then we'll discuss visualizing sample relationships in beta diversity called ordination, and discuss the arch and horseshoe effects, which are common artifacts in ordination. Then finally, I'll briefly cover some new methods for addressing challenges in beta diversity analyses. So to provide some context of beta diversity analysis, let's consider an abundance matrix visualized here on the left, where we have samples in the rows and OTUs in the columns. You may notice that the middle three columns are the same between all samples, which we could call redundant information between samples. So beta diversity analysis intends to explore and recover non-redundant variation between samples. So we're not assuming all of these samples are necessarily very alike just because they have some redundant information. So we're highlighting that variation in subsequent ordination shown on the right. Current challenges in beta diversity include study design and sample sizes, which are obviously limited by the costs and time imposed on researchers, and this has a large impact on statistical testing of beta diversity analysis. We may also discuss extraction and sequencing artifacts, which are a common problem for beta diversity, coming from batch effects, read depths, and sequencing errors. But this has been discussed already at length, for instance, in Glor et al. From 2017. And then there's also a lack of standardization in beta diversity analysis. So there's inconsistency of which pairwise measures are used or ordination methods used, which can in turn lead to difficulty comparing studies that are using different methods between them. Subsequently, biological interpretation of these respective methods may be limited as we have an incomplete picture with beta diversity analyses, but new methods may be able to give us more and more information. For instance, the work that's being done in creating more functional annotation. So today I'll be focusing primarily on the lack of standardization and then biological interpretation for beta diversity, as the prior two challenges have been discussed at length and are still ongoing. So I conducted a literature review of 150 microbiome studies published between 2017 and 2022 in order to identify the popularity of different beta diversity measures. And far and away the most popular were Bray-Curtis and Unifrac. Unifrac distances, both weighted and unweighted, are combined here, so I didn't split those up. What was also interesting about this, though, was that most often a microbiome article only reports one beta diversity measure, despite there being many, many measures that can be used. Some studies obviously did report more than one measure, but it was rare to see more than one and rarer to see more than two. Beta diversity measures in their multitude can be identified in two main groups, distances and dissimilarities. And to just briefly cover why some are distances, these distances must meet the requirements of a distance metric, which is fourfold. First being that the distance measures must be non-negative. Secondly, the same 2 points must have a distance of 0. So if 2 points are the same, then they must be a distance of 0 from each other. Symmetry, which means that in a pairwise distance, the order of the respective points does not matter. The distance outcome is the same. And finally, it must meet the triangle inequality. So example distances are Unifrac, Euclidean, which is Cartesian distances, chi-square, and Aitchison distances, which are chi-square and CLR transformations prior to Euclidean distance. And finally, dissimilarities are going to meet some, but maybe not all of these distance metric requirements. And those could include Bray-Curtis, Jacquard, or cosine. For a discussion, I'll be using the human gut microbiome dataset of US immigration from Thailand. So this followed Hmong and Karen individuals and compared to US-born European Americans. And for the sake of our analyses, I excluded the first-generation individuals, the Hmong first generation and Karen first generation, due to the large degree of dispersion in these groups, just to simplify analysis. So using this immigration human gut microbiome data set, we can compare a large number of measures, and this reveals associations between said measures. So if we're using the microbiome, if we're using a rarefied table from the immigration microbiome data set, then we're coloring based on the pairwise value, whether it was within an ethnic group, that is, if two samples are a Hmong second generation sample, then it would be colored orange for within ethnic group. And if it's outside ethnic group, then it would be colored purple. So purple distances, obviously we would expect to be larger than the orange within ethnic group distances, because the study did find that most of the time the ethnic groups were clustered together. So this plot is very overwhelming, but if we look at just some of the distances or dissimilarities that are well correlated with one another, the top left corner sticks out the most. So looking closer at these, we find that Bray-Curtis, Kolczynski, and Manhattan distances are functionally equivalent when we're using rarefied data. To briefly describe why this is, the numerators of these respective measures are functionally equivalent, and then the denominators of Bray-Curtis and Kolczynski in particular are accounting for sample size, which in a rarefied data set are all the same. So that's why these three are all functionally equivalent here. And then the cosine distance dissimilarity is strongly correlated as well, interestingly enough. To guarantee that this is still the case if we're not working with a normalized data set, I borrowed the soil data set from Lauber et al. And looking at normalized OTUs, we see the same relationship as found in rarefied data. And then with raw counts, we find that Manhattan in particular no longer looks like the Bray-Curtis, Kolczynski, or cosine because it is not accounting for sample sizes in its equation. So an important consideration when there's no normalization or rarefaction. Another interesting relationship comes up between the unweighted measures of Jacquard and unweighted unifrac. So taking a closer look again, we see that these are very strongly correlated, which is unexpected given that unweighted unifrac is supposed to be incorporating phylogenetic information from a tree. So this would imply that the unweighted nature of the distance metric unweighted unifrac is actually more important to the distance's outcome than the phylogenetic information, as Jacquard and unweighted unifrac are correlated. Accounting for compositionality can take on many forms. For instance, the chi-square distance is a chi-square transform, but it's still going to have a squared term the same as Euclidean, which in turn we find in the measure of distances is going to particularly focus on outliers and exaggerate perhaps the distances between certain outliers. So that's what's happening with this small cloud you'll see in the Euclidean rows and columns, where that upon further analysis was just one sample that happened to be further from the rest of the group and was exaggerated by Euclidean distance. Aitchison and robust Aitchison distances are accounting for compositionality with the centered log ratio CLR transform. And robust Aitchison distance is actually incorporating matrix completion so that you can fill in all of the zeros in your sparse microbiome data set, rather than just adding some arbitrary value as you would for Aitchison distance, since CLR transforms are non-compatible with zeros in your data. But they are not significantly correlated with one another, so it's important to note that this matrix completion makes the outcome distances very distinct from Aitchison. So we find that we can compare within groups of measures, but between groups of measures, we have to really consider the interpretation. And these respective groups are all going to have very different interpretation, as well as if unifrac distance is included with phylogenetic information. But between, for instance, Bray-Curtis and Jacquard in two studies, you could identify that if Jacquard was found to be particularly significant compared to Bray-Curtis, then the simple presence of a set of OTUs is impactful for distinguishing between samples. Visualization and ordination poses another level of complexity in beta diversity analyses. And we found that far and away the most popular is PCA or PICO-A, which we grouped together. And this makes sense because it is highly reproducible using matrix decomposition. Next most popular was NMDS, but this measure uses ranks of distances, so it can be helpful in that it's not necessarily using the harsh distances, but rather a rank, so it can be more compatible with exaggeratory distances like Euclidean distance, and it iteratively fits samples to a reduced space. Also, growing in popularity is TS&E, which is assuming an underlying manifold but can overemphasize local optima due to gradient descent. And finally, RDA, which is supervised and incorporating metadata, but could be disadvantageous by its supervised nature. Procrustes analysis shows us that all of these are significantly distinct from one another, so we can't necessarily compare the positioning of samples in one plot to the positioning of samples in another, as they are distinct, and we have to instead interpret the underlying algorithm. New approaches for visualization include deep micro, the autoencoder, and other autoencoder approaches. As machine learning is rising in popularity, we find more and more autoencoders in the beta diversity space, and this makes sense because in many machine learning applications, autoencoders have been found to outperform PCA and PICO-A, where those were previously the standard. However, autoencoders may be difficult for interpretation as they are more of a black box algorithm, so a shallow autoencoder may be advantageous because it's more interpretable. Finally, we've discussed briefly the arch and horseshoe effects in visualization, which is a common artifact of ordination where the samples seem to form an arch. In this particular study, the soil data set from before, we find that the pH scale along which the samples were taken is forming an arch in the data set, irregardless of what distance or dissimilarity measure is used. So our lab came up with a solution called LMDist, which resolves these arch geometries. So in this particular data set with soil pH, we find that the pH gradient is resolved along the first dimension when we adjust distances. So LMDist is not itself a distance metric, but is more of an adjustment of distance measures, because we found that distance measures become oversaturated and form these arches or horseshoes. And most importantly, we actually find that other variation is then resolved along the second dimension when you resolve the gradient along the first. And so annual season temperature became significant after applying LMDist. And LMDist is able to resolve gradients in both other real data sets, such as the Guerrero-Negro microbial mat data set, which is sampling microbial mats at varying depths. And then in simulated data sets, such as a simulated gradient data set we created, and then the somewhat famous Swiss roll from manifold learning. So to overview, considerations for current challenges are the lack of standardization. We want to compare measures that are within groups of measures. And if they're between groups of measures, then you have to understand the interpretation of comparing what one measure might be accounting for versus another measure. And then autoencoders are a popular and efficient approach to apply machine learning to beta diversity visualization. And arches and horseshoes appearing in visualization can be adjusted with LM-dist, and that's compatible with any distance or dissimilarity measure. Biological interpretation, once again, comes down to understanding the underlying impacts of different measures, and you can interpret even more by using more and more measures or more and more ordination methods. Finally, thank you to my colleagues, and thank you all for joining me.